Got a big biology exam coming up? Good. Let's spend the next 5 minutes absolutely locking in the concepts of restriction enzymes and gel electrophoresis. Think of this as your ultimate last-minute study session. Let's get right into it. Okay, think about it like this. You're standing in a library with thousands and thousands of folks, and you have to find one specific sentence. That sounds impossible, right? Well, the human genome is kind of like that library. It's got about 3 billion letters of code. So how on earth do scientists find and work with just one tiny, specific piece of that giant puzzle? They need a tool, a really, really precise one. And that tool, the answer to our library problem, is an amazing group of proteins that act like, well, molecular scissors. They are the absolute key to making precise, predictable cuts in a huge strand of DNA. So let's get the official definition down. They're called restriction enzymes. And their entire job, their whole purpose in life, is to scan along a DNA molecule, find a very specific sequence of DNA's building blocks, those are called nucleotides, and then snip, cut the DNA right at that spot. Now, what's so incredible here is just how specific these enzymes are. Take a look at this example, E-CoRI. It is programmed to find one sequence and one sequence only, GAATTC. It will ignore everything else. Once it locks onto its target, it makes the cut. It's not random at all. It's like a tiny biological guided missile. But it's not just that they cut DNA. The really cool part is how they cut it. The type of cut they make is what opens the door for genetic engineering. And this brings us to a super important concept you are definitely going to see on your exam. See, the real magic, the thing that makes these enzymes the foundation of modern biotech, is that the cut usually isn't a clean, straight slice. It actually leaves a little piece of DNA dangling, an overhang that isn't paired up with a partner. And this image shows it perfectly. These single-stranded overhangs are called sticky ends. Why? Well, because they're chemically sticky. They really want to find and bond with a matching complementary piece of DNA. So if you cut another strand of DNA with the exact same enzyme, it'll have a matching sticky end. You can then basically glue them together. This is the whole basis for creating what we call recombinant DNA. Okay, so we've used our molecular scissors. We've chopped up our DNA into a whole bunch of different sized pieces. Great. But now what? We've just got this jumble of fragments. How do we sort them out so we can actually study them? That's where our next amazing tool comes into play. And that technique is called gel electrophoresis. Really, all it is a way to sort molecules. For us, it's how we sort all those DNA fragments we just cut up. All you need are two main things, a slab of gel and an electric current. The absolute best way to picture this is like a race through a thick forest or maybe an obstacle course. The gel is like a mesh with tiny pores. As you can see here, the small, nimble DNA fragments can zip right through those pores and travel really fast. But the big, bulky fragments, they get tangled up, they get slowed down, and they just can't travel as far. So let's just quickly walk through the procedure. First, you cut up your DNA with those restriction enzymes. Second, you load those little fragments into tiny pockets called wells at one end of the gel. Third, you flip the switch and run an electric current through it. Now, here is a key fact you have to remember. DNA has a negative charge. And what do we know about charges? Opposites attract. So that negatively charged DNA gets pulled through the gel towards the positive end. And ta-da! Here's what you get at the end. After you turn the power off, you see this pattern of bands. Each band is just a pile of DNA fragments that are all the same size. The ones at the very bottom, the ones that travel the farthest from the starting line, those are your smallest, fastest fragments. The ones up top, closer to where they started, those are the big, slow ones. So we can cut DNA precisely, and we can sort the pieces by size. Why is this such a big deal? Well, when you combine these two powerful techniques, you get the foundation for one of the most famous tools in biology, DNA fingerprinting. So here's the secret sauce. Our DNA is packed with these little regions of short repeating sequences. Think of them like stutters in the code. Now, the crucial part is that the number of times these sequences repeat is different for pretty much everyone. That variation is exactly what makes your DNA profile unique to you.
Okay, pay close attention because this slide brings it all home. Scientists design the restriction enzymes to cut on either side of these repeating areas. So if you have more repeats, like the person here with 12 and 14 repeats, you get a bigger chunk of DNA. If you have fewer repeats, you get a smaller chunk. Then, when you run them on the gel, those different sized chunks travel different distances, creating a totally unique pattern of bands for each person. And that is a DNA fingerprint.
Whew, okay, we've covered a lot of ground, so let's boil it all down to the absolute essentials you need to nail on your test. This is the moment to get that screenshot finger ready. All right, let's do a rapid-fire review. Restriction enzymes, they're your molecular scissors. They make sticky ends so you can paste DNA together. Gel electrophoresis, it sorts DNA by size using electricity. Why does that work? Because DNA is negatively charged. And always remember, smaller means faster and farther. Put it all together, and what do you get? DNA fingerprinting. You got this.
So we'll end on this thought. We now have these incredible tools that let us take DNA apart, like a set of Legos, and then sort all the different blocks by size. Which leads to a really mind-blowing question. Now that scientists can so precisely deconstruct the blueprint of life, what could they build with it? That's a story for another time. Good luck on your exam.